The next round of talks is really going to start to focus in on analyzing proteomics data. And so Yuqian is going to kick us off here again, talking about targeted proteomics pipelines. Yuqian?
Yeah, here. Can you see my slides?
We can. Thank you.
Okay. So, so in this talk, we will talk a lot of the details of how to set up the acquisitions in discovery and the proteomics, targeted proteomics data pipeline, and how to do the data analysis. So we will start. So for data acquisition in discovery proteomics, there are specific considerations to both liquid chromatography and mass spectrometry. So when setting up experiments for liquid chromatography, the first thing is to decide which type of column particles you want to use, especially how big the particle size. So in modern liquid chromatography technique, the columns that packed with less than 5 micron particles are called high-performance liquid chromatography, while columns packed with sub 2 micron particles are called ultra-high-performance liquid chromatography. The smaller the particle size, the better the separation power. So you will have more peptides ionized than detected by mass spec. Because of the better resolving power, you can run the system faster with higher throughput. However, the back pressure of ultra high performance liquid chromatography is super high. You need more power to push the liquid to get by smaller particles. The most advanced ultra high performance liquid chromatography can hold up pressure to 15,000 psi, which is equivalent to 15,000 pounds per square inch. So both 5 micron and 1.7 micron particles are commonly used for proteomics studies. You need to also decide on the length of the column and the column diameters. The longer the column, the better the separation. The smaller the diameter, the better the ionization efficiency for electron spray ionization. And once you have the column, then you can decide on the gradient. So gradient really means like the ranking up of organic contents in your liquid that are going through the column. The longer it takes for the organic percentage to go up, the better the separation. So normally we use 1 to 3 hour gradient for discovery proteomics analysis. To set up the mass spec for discovery proteomics, you will need to decide on the resolution of mass to charge ratio and how much ions you want to assign to the orbitrap, for instance. So in an ideal case, you want high resolution and lots of ions, but there is always some trade-off between the resolution and the number of ions with the speed. So you can now go fast if you use you cannot go very fast if you use very high resolution and sending in lots of ions. So, but if you are slow, you might miss some peptides since the liquid chromatography cannot wait because it is continuing eluting peptides out. So you need to be fast. So you will also need to decide how much exciting energy you put to fragment the molecular ions. Also, you need to set up like how many molecular ions to be fragmented when you see it in the MS1 spectrum. So we will talk a little bit more about how it actually gets done to select the top most intense ions. So in mass spectrometry, the instrument runs in scans. So for example, the first scan you will have a MS1 spectrum acquired. And inside the spectrum, you have lots of molecular ions. You can select the top 5 most intense ions. So you will do fragmentation of each of them in the next 5 scans. So scans 2, 3, 6 will be MS2 scans, and molecular ions are selected based on the MS1 spectrum. And then after you go through all the 2, 3, 6, you will go back to do MS1 again. So in this case, like if your MS1, the second MS1 you get is similar to the first one. So normally, because there are so many ions we want to see in this, so many peptides we want to fragment in this MS1 spectrum, you will normally exclude the ions you already fragmented in the last, for example, last 30 seconds. So in this case, like we will do some other, like we will fragment some other molecular ions, which we have not done yet. So if we already done it, we will not do that. So in the next set, you will do a different set of ions for the next 5 scans of MS2 scan. So this is why it's really not ideal to use spectral count for quantification, because it depends, the spectra is really depending on the MS1 spectra, like the intensity relevance between peptides in the MS1 spectra and what you get from the MS1 spectra. So nowadays, the mass spectrometry is super fast. You normally can pick like top 10 or even more, like from the MS1 spectra to do the MS2 scan. So this is what we'll get like in the scan order. So you will have MS1, lots of MS2, MS1, lots of MS2. So in data analysis, there are a lot of things we need to consider. The first one is we want to assign the MS2 spectra to peptides in the database. So every fragment ions you see here, for example, this one, for instance, if you look for the details, it's actually has a distribution of isotope. And we need to convert those from a wide distribution of peaks to a centralized centroid data. So you have just one single value. And then you need to de-isotope it. So you have single value for fragment ion. And then you do this for all of those fragment ions, and you get a list of monoisotopic masses. And then you can use that to match what you get from the peptide library. So in the peptide library, you have also a list of potential theoretical monoisotopic masses from peptide, and you match those together. And if you find a good match, so it is a peptide spectral match. And also, when you search the database, you need to consider some modifications. For example, we normally alkylated the cysteine, so you need to add alkylation, the actual mass, to the cysteine residue. Also, it's very possible for methionine to get oxidized during the sample preparation process. So you need to add those to the database search. But because the oxidation of methionine is random, so you can set it at a variable search. That means it can be there, it does not need to be there. But for the cysteine ones, because you specifically label it, so you can set it, you need to set it to the fixed modification. And most importantly, as we mentioned before, if you are not enriching your PTMs, post-translational modifications in your sample preparation steps, it is very unlikely you will see them in your data. So because of that, you do not add those PTMs in your search. If you add them, it's just stretch your search engine. It takes longer to search, but in the end, you still don't have anything for the PTM. So that is one thing to consider. Also for the peptide to spectral matches, there are some good matches and some bad matches. So for example, in this left figure, it's a very good match. It will have a higher quality score. And in the right, it is a lower quality match. And so when a spectrum match to multiple peptides in the proteome, the software will pick the peptide that scored the best as it assigned the peptide. The exact scoring algorithm vary greatly between softwares. Also, just as any instrument, there is some variations in the mass accuracy. The mass accuracy is measured as like a precursor m over z minus theoretical m over z of the peptide and divided by the theoretical. So you get a ppm error, and this error can vary, depends on the time you acquire the mass spectra. It can also vary by the m over z of the peptide. So you need to do a refinery to refine the distribution of this mass accuracy to set it to around 0. So this is, here is the original one. After refinery, you will have a distribution around 0. And after this refinement, you can set mass accuracy filtering criteria to remove any peptide spectral matches where the theoretical and the predicted method are far off, like the tailed end of this distribution. Also, since we cannot look at all the spectra one by one in this kind of large data size, so what metric we can use for global confidence assessment of a large-scale proteomics data? So we typically use a target decoy strategy where we employ like a score threshold for achieving mass false discovery rates at a desired level. So the most widely employed methods for generating decoy database are reversing the original targeted database, or you do the randomization of your original database. So you can choose either of those as your decoy library. So you combine the true ones with the false ones together to search against your MS2 spectral library. And in the end, you have some matches from the true peptides and also some from the false peptides. And then you can calculate the false discovery rate, which you can either use, either of these equations. Normally we use at least 1% FDR to filter out our data. So the 1% FDR will mean like, for example, we have 100 peptide spectrum matches. There will be one which is a false match. So there are many softwares can be used for discovery data analysis. They all need to start with the raw files from the mass spectrometer and the FASTA files containing all the protein sequences in the organism. And in the end, you will get some tables of peptide spectral match. And also you will get like the MS1 quantification, like an intensity value for the peptide identification. One of the software set is MSGF and the MASIC, which are two PNL softwares. You can download them from like a PNL website. They provide excellent identifications of peptides from the mass spectrometer. Especially MSGF can significantly increase the number of identified peptides compared with commonly used methods for peptide identifications. Another one is MaxQuant, which is it's kind of a software suite. So you can do a lot of things with it. It's kind of an integrated tool. You can do search, and they can do FDR control, a protein roll up. You can also visualize your data. So it is all in one, but it's kind of like a black box. And it is widely used in scientific community. So in the tutorial science session in the afternoon, we will talk about these two softwares. Now we will talk about the targeted proteomics pipeline. So targeted proteomics, especially the selected reaction monitoring, or so-called multiple reaction monitoring approach. They use a triple quadrupole mass spectrometer. So there are 3 quadrupoles, and the first one, third one are selecting ions, and the second one is fragmenting ions. And selection of the precursor, which is molecular ions, and the fragment ion pair, which we call transition in SRM-based targeted proteomics. And compared to discovery proteomics, SRM has better selectivity, sensitivity, and wider dynamic range because it is not using the scanning mode. The SRM is just continuously monitoring the transitions. The SRM is a method of choice for accurate quantification of protein expression levels in the biological system. So the first step in targeted proteomics is really to select the protein targets. You can select those from your discovery proteomics data, and some people see some significantly differentiating proteins in their samples, and we want to validate whether it is true. They do SRM for more accurate quantification. Also, you can, if you are doing like a synthetic biologic work, you already know your pathways. You can just choose the proteins in your pathways on network of interest. Or sometimes you can find some interesting proteins from the literature. Basically, you just need to know which peptide, which protein you want to do targeted proteomics before we can start. And once we have the list of proteins, target proteomics assay need to be developed before you can take the full advantage of the quantification. So first, you will need to select peptides. You will need to find some peptides which can fly well, and it's a true representation of the protein abundance. Also, you need to decide which transitions to monitor in your assay. And finally, you will have a ready-to-go assay in the end. So how we can select best performing peptides? There are some criteria. The peptides should fly well. So normally you choose peptides that has higher spectral counts in your discovery proteomics data. The peptides that has a higher spectral count normally means they are very easily to be detected in mass spec. Also, if you don't have the discovery data, you can still choose peptides based on some theoretical prediction scores. So here are some choices for you to use to get the prediction scores. And also the SRM peptides cannot be too long or too short. So short peptides can have lots of interferences in quantification. And the long peptides, most of the time, they don't fragment well. And also they are hard to synthesize. So normally like we choose peptide between 6 to 25 of amino acid. And also peptide needs to be unique to the protein. Otherwise, it's not a true representation of the protein. So there should be like a one-to-one match between the peptide and the protein. Also, you need to make sure like your peptides are fully tryptic. But because like a missed cleavage can be a common problem in protein digestion. So but missed cleavage size are hard to predict. And they are not, in abundance-wise, they are not very stable. So you try to avoid as much potential miscleavage as possible. So there are some criteria, like some peptide like amino acid motifs you can avoid. And also, you try to avoid as much as possible some potential post-translational modifications. For example, methionine can be oxidized, N-terminal Q can be cyclized, and some other stuff to consider. So after you have the peptide, you can purchase either crude or pure peptide. It just depends on the purpose of your experiment. If you want to have an absolute quantification, you can purchase the pure peptide. Otherwise, you can go with the crude peptide, which is much cheaper. So once you have the peptide decided, you also need to select transitions for mass spec to monitor. So remember in the last talk that commonly multiple transitions were monitored for each peptide. So they are selected from the fragment spectra. You will want to have the transitions that have high signal, like Y9 and Y8. There are also some things you need to consider, you normally we only select Y ions, since B ions are often of low abundance and absent in triple quadruple. And also you never select Y2 and Y1 ions. Y2 and Y are basically just one amino acid or two amino acid, which is very short, has a very low M/Z, which can have lots of interference from the mass spectra. You don't want to also have M/Z too close to the precursor ions. In that case, you will get a huge peak because the precursor is abundant. Also, the higher the mass, the better. So you can select Y10 in this case, which is higher. So after you have the transitions, when you do the final assay, you also need to select peptides which are actually detected. So in these two figures, the left one, peptide A, is detected. As you can see, the top is the endogenous signal for the peptide, and the bottom is internal standard signal for the peptide. And there is good matches between the ratios of transitions in these two cases for the endogenous and the internal standard. So it is a good match. And then also they elute together, and the intensity of the endogenous is good. So this is a good peptide which is detected. But on the left and the right is a peptide which is not detected. You see some peaks of the endogenous, but it's embedded with the noises. So it's at a noise level. So you are not sure whether it's detected or not. So this B peptide, you will not be selected in your final assay. Also, if you have multiple peptides, you can choose from, and you can do a correlation plot between different peptides. You can see which peptides correlate well. For example, this X peptide and the Y peptide, they correlate pretty well, has a score of over 0.9, but they don't correlate well with this peptide Z. So there could be some problem with this peptide Z, which can be having some unknown modifications on it, something you don't know about it. So when you decide on the final assay, you will definitely delete this peptide Z, choosing only the peptide X and the peptide Y. So, so now you have finalized the assay of peptide and the transitions to monitor. And when you do the acquisition on the instrument, a lot of times you will have thousands of transitions to monitor in one single analysis. Because the instrument cannot take that many transitions all at once. So you need to schedule properly your transitions along the retention time. Like if you see here, if you set your transitions to like the scheduling window to 7 or 10, it increased the number of concurrent transitions. But if you set it very small, it decreased the concurrent transitions. Yeah, so that is also something you need to consider when you set up your data acquisition in SRM. So when you have the data already with the SRM, you can import it into Skyline, which is freely available and open source software from developed by University of Washington, and you can use it to visualize your data and to extract the peak and the curve for your as your intensity abundance for your endogenous and the heavy, and you can do a lot of things with it. So we will talk some more about how to navigate Skyline in the afternoon tutorial session. So in the final part of this talk, I will show how we can use targeted proteomics to quantify protein expressions in agile biofoundry projects. So, and also how we can improve the throughput of the design, build, test, learn cycle for the synthetic biological work. So in this work, we studied 132 enzymes in the key metabolic pathways in the Pseudomonas putita. And we selected 339 peptides for the proteins. And we synthesized the internal standard and spec them into the samples, and I run them through the targeted proteomics assay. And to improve the throughput, we utilize a large column, which is called microflow LC-SRM instead of nanoflow LC-SRM. Both of these columns use the same particles, so they have same resolution. The first one is bigger, and the second one is much smaller. Because of that, you need to load more samples onto it. But normally it's not we have plenty of samples in synthetic biology work, so it's not a problem. And the gradient was greatly shortened to 30 minutes. Instead of like the nanoflow, we use a 2-hour gradient. So this can increase the speed of analysis by 4 times, while we still maintain the sensitivity of measuring a quantification, as demonstrated by the limit of detection and the limit of quantification. So the limit of detection and the limit of quantification are quite similar between these two setups. So we applied this approach to test multiple samples of Pseudomonas putita, which are grown in various media conditions and with different food sources. And this is the significantly differentiated proteins comparing between conditions. For example, in figure A, there are slight differences of enzyme expression levels between glucose-fed samples grown in MOPS media versus those in M9 media. And the major variant enzymes between the two conditions are the ones in the TCA cycle, EMP cycle, and the PP cycle, PP pathway. And also we can compare between different food sources in the same media. This is comparing between glucose and coumarate. This is comparing between glucose and fructose and glucose versus gluconate. So we can do a lot of things with accurate abundance comparison between samples. For example, this one we use like a fold change of 2 as cutoff. So we can be sure like our two-fold change is really the very significant in when we working with targeted proteomics data. And also we can look at the fructose and the glucose and the glutamate metabolisms, and also the coumarate metabolisms through the TCA cycle. What are the differences in the proteins abundances if we're using different food sources? So that's actually the end of my talk.
That's great, Yuqian. I think it gives a lot of detail that you typically don't see with a proteomic talk. So that was fantastic. I wrote down some of the questions that I've been seeing come in that I can verbally tell you if you want to answer them, if that's easier. So for discovery proteomics, one of the question was, how many peptide spectrum matches or PSMs are typically required for a protein assignment? So how many peptides do you usually require for a confident protein identification?
Normally you can do 2, like 2 peptide. If you found 2 peptides from the same protein, you can be more sure like you identified this protein.
Great. There was another one for discovery proteomics, and it had to do with your refinement of mass accuracy. And the question was, is it performed before database searching? So is it within the tools you're using like MaxQuant or MSGF, or are you doing it with another tool before it feeds into those pipelines?
So to get the original one, you need to have a search. So normally they do 2 search. So first one you do a search and you get the initial original profile, and then you do the refinement, and then you search again. So this is all enclosed in, for example, the MaxQuant. You don't need to set up by yourself.
Perfect. And then there were some great targeted proteomic questions. So one about, so you mentioned the expensive absolute heavy peptides, and there's another one for the crude peptides. So do you feel like if you use the crude peptides, does it still give you accurate quantification?
Yes, not accurate as in the sense of absolute. So you don't know how much exactly in the sample, but it's still very good to quantify between samples, between conditions. Yeah.
Great. And here's another targeted one. So when doing targeted SRM or MRM, are there additional sample enrichment or purification steps before adding the isotopically labeled standards, or can you just run a global triptych digest?
Can you repeat?
Yeah, so I think, so they're pretty much asking, so when you do, when you're running the answer, so when you're doing the targeted SRM or MRM, do you need to do any second enrichment steps, or can you just take your crude peptide extract, put in your isotopically heavy peptides and just run it?
So I think the actual enrichment or extraction step can be applied to both discovery and targeted. So discovery targeted is really how you run your instrument. So if you want to have, for example, in the plasma samples in human, if you want to get rid of the albumins and stuff like that, which are very high in abundance, and you don't want to monitor those, and you can get rid of those by like enrichment columns or like depletion columns. You can do that. You can do that for both discovery and targeted. And you normally do those kind of steps before you add any spec-in peptides. And you can also do like a phosphorylation with targeted. So you just enrich the phosphopeptides and add the phosphorylated internal standard and do the targeted. You can also do that. So really the spec-in is happening before the LC.
So enrichment can help in all cases then,
Yeah,
but you don't necessarily have to do it. Because I remember you even did some targeted analyses when you were looking at soils and things like that.
Yes, yeah.
So yeah, either can work great. It looks like there's another question that just came in on Zoom. It's for your last, so for your paper that you were showing, for the example shown at the end, did you combine different groups of data to draw the volcano plots? How was that done?
So to be able to get a volcano plot, you need to statistically compare your proteins between groups conditions. So you need to have like a fold change. So how much fold change and what is the p-value, like confidence value, and then you can plot. It's just like a regular scatter plot. You can plot like x-axis is your like fold change, y-axis is your p-value. But before that, you need to have a statistical analysis, which is going to be done on maybe the
Thursday.
Thursday.
Yeah, which is and you sent all of your data through PMART, so it's going to be a lot of the things that are covered on Thursday are the things that you used for your paper too, correct?
Yeah.
Oh, here's another one. Do you use the software to select the peptides to analyze in MS1, MS2, etc.?
The software comes with the, if you are talking about the discovery, I think the software comes with the instrument. Like if Thermo has the instrument, they will make a software like along with the instrument, they can run like do it when they acquire the data. They select peptides from the MS1. So this is comes with the instrument. The mass spec instrument should have those functions.
Perfect.
And what you can do to set up like how many top 10 or top 12 or something. But the instrument will do that work for you.
Yeah, and one more question about the volcano plots. I have this feeling it was your own R scripts, but what platform was used to generate the volcano plots? Is it Skyline?
Skyline cannot do that. So as I mentioned, the volcano plots, before you do that, you need to have statistically validated and compare the data. So you have some like fold change and p-values. And to plot it is very simple. You can even plot it in Excel. So, but you need to have some statistical analysis to be done.
Yeah, and I think what Yuqian was showing today is there's so many different software programs, search algorithms, all these things are applied and they're very complex. And what you get out of it a lot of times is a simplistic table where you have your identifier, your peptide, and then you have your data set, and then you have your relative or absolute abundance. So once you get that confident data, you have a lot of different options on how to do the statistics, and then how to plot the data, how to model the data. So
yeah, that's really on the Thursday.
yeah. I hope I didn't miss any. I think I got them all.
And David has put a link to the PMART, information about PMART, that statistical analysis, in the Discord in case anyone wanted to look at it sort of ahead of time, ahead of Thursday, since there seems to be interest.
Yeah, and the nice thing about PMART is, it has flexibility in the type of data that you can put into it. So that's why it's following after the metabolomics data tomorrow, because you can use it for proteomic data, metabolomic data. And I think someone also, had a question about the pre-processing and data cleaning type things. And for normalization, looking for outliers, that is going to be on Thursday. So that's kind of part of that pre-processing that goes on. Okay, I think we have about 5 minutes. Is that right? until?
Yep. We do have a few
Yvette.
extra minutes before Yvette, in case there was another question.
I guess I can ask a general run. So when you're doing your synthetic biology analyses, Yuqian, you know, when you first start out, you have a brand new organism, you know, would you choose discovery then versus targeted because you haven't done that many analyses before, and then move to more of a targeted approach later to make, because it's going to be faster and high throughput and you're going to know more about your data?
Yeah, especially like there is a step in targeted, you need to select peptides. So it's very good to run some discovery first. So you know like what peptides you are seeing in the system. Yeah. And at the same time, discovery can give you some like quantification to the samples you are running. So normally like people just first run discovery and see what's in there. And also because I targeted, it takes a lot of time to do the assay development. So yeah, it's easier to just run it on the instrument with discovery.
That makes sense. So both are very useful. Oh, and we have one more question in Zoom. So Yuqian, do you have any recommendation for data visualization software for discovery proteomics data?
For visualization, if you just want to see the spectrum, like you, for example, MaxQuant can do that within its software. And David is going to introduce another one developed at PNL. You can also visualize mass spectra. If that is what you mean.
Yeah, because there's lots of different ways to visualize it. Okay, and there's also a comment saying that there was a question on MPlex extraction efficiency on Discord that they were hoping for us to answer. Let's see if I can find it. Okay, so there's two. So MPlex was our multi-extraction, and it asks if we've done it on mammalian cell cultures and what about plant cells. So for that one, mammalian cell cultures, those are very easily lysed. Almost the solvent extraction itself will separate it, and then you can get your different layers. With plant cells, yes, it does work, but you might have to, just like we did with the fungus cells, do some extra grinding because they're going to be harder to lyse. And how efficient is MPlex extracting transmembrane or membrane proteins? How can we account for differences in extraction efficiency of membrane slash soluble intracellular, extracellular when interpreting protein abundances? So a lot of times people say with mass spectrometry that we have it's very challenging to say transmembrane proteins, especially those peptides that are, in that hydrophobic region, without, we actually see quite a few of them. So Yuqian, when she's done the discovery and the targeted analyses, I mean, we usually actually detect quite a few membrane proteins, and a lot of the membrane proteins we detect are by the peptides that are on the outside of the cell. So, it might be, we're still detecting the membrane proteins, but we're using peptides that we observe very well with mass spectrometry. So I think, different techniques can do a really good job targeting membrane proteins, but we do see them well in our Agile Biofoundry analyses too. Do you have anything to add, Yuqian, about transmembrane proteins?
I think what you said is good. Yeah, we do see some of them, like they are transporters, especially. Yeah.
Yep, and they're important for the 3HP studies we did,
Yeah.
figuring out how that's getting extracellular. So surprisingly, we're getting really nice coverage for those. Okay, I think it's Yvette's turn.